So, chromatography. Chromatography is a separation technique. We know that because we've looked at this previously. Syllabus point that we're looking at is this big long one here. It can be used to determine the composition and purity of substances, and the important part from what we're going to discuss a lot of today is the separation of the components is caused by the variation in strength of the interaction between atoms, molecules or ions in the mobility and stationary phases. So we're dealing with intermolecular forces when we're talking about chromatography. So chromatography is the technique where we separate out the different components of a mixture, and we separate it out as a result of differential distribution between the stationary phase and the mobile phase. So the stationary phase is in what you would have done previously would be the filter paper, and the mobile phase will be the solvent. So we need to know how the solute, which is what we're trying to separate, dissolves in the solvent and how it interacts with the material that we're, the stationary phase, what we're trying to do the chromatography through. There are three different types of chromatography that we're going to talk about. There's thin layer chromatography, gas chromatography, and high performance liquid chromatography. So thin layer chromatography is what you would have done in past years if you've done any chromatography. So a bit of terminology. It works on the basis that different materials will absorb onto the surface and desorb into the solvent at different rates. So when we have our mixture, which is made up of different materials or different substances, those different substances will absorb, stick to the surface, and desorb or dissolve into the solvent at different rates. And as a result of them doing that at different rates, they move up the stationary phase at different distances over time. So what we can do is we can alter the mobile phase and the stationary phase or the surface, so we can alter the solvent and what we're doing the chromatography through, which will allow us to separate the different mixtures. So some things that will affect how far and how fast the material or the substance being separated will move, which is called the analyte, is the different types of polar groups. So we've been discussing that. The amount of charged and polar chemical groups present, the molecular weight, its geometry, and the position and numbers of carbon-carbon double bonds. They all influence how fast or how far the material being analysed will move through the stationary phase. So, thin layer chromatography. It's the simplest, quickest, easiest way to do things. Not necessarily the most robust way, but it's the quickest way to do it. Often used to analyse the presence of drugs or amino acids in samples. It's faster and often provides better separation than paper chromatography. So we would have done paper chromatography in the past. So you have a plate, which is your stationary phase, which contains absorbent material bonded to either a glass or a plastic support. So in the photo here we've got an example of the stationary phase that we're using in thin layer chromatography. And the idea is that it provides a very large surface area, so there's lots of opportunity for the different phases to either be absorbed to be absorbed by the stationary phase. So what we do is we draw an origin line at the bottom, which doesn't go into the solvent, and that's the line on which we place the sample that we're going to test. So you would have done this last year when you did the DNA separation, and you would have drawn your line on your filter paper and that's where you would have placed your extracted DNA. So separation occurs as a result of the solvent moving up the stationary phase, and it moves up there as a result of capillary action. So when you're using paper, filter paper, then it just simply will move up the filter paper as a result of capillary action. So it moves up towards the top of the plate and you get a solvent front. So the solvent front is the maximum extent that the solvent has moved to. So if you had something that was perfectly dissolved in the solvent and wasn't being adsorbed to the stationary phase, then the solvent front is where you would end up with your analyte. But that generally won't happen. So it moves up. So in this case here, we've got a yellow dot, a blue dot, a green dot, and a red dot. The yellow dot has moved the least distance. The red dot's moved the furthest distance. You can also see that it's spread out the most. And they have moved different distances as a result of the different composition of those substances. So samples that will desorb into the solvent, so they dissolve really well in the solvent, will move up the plate quickly. Whereas samples or substances that are absorbed by the stationary phase will move up and so therefore are not dissolved in the solvent as well, will move up the plate much slower. So this one here would be one that was more attracted to the stationary phase. This one here is more attracted to the mobile phase. So it really comes down to the idea that it's a competition of attraction. So if the material is attracted more to the stationary phase, then it doesn't move up as quickly. If it's attracted to the solvent or dissolves in the solvent more, then it will move up more quickly. So you then measure the distance that it's moved, and then you divide it by the distance that the solvent has moved. So you've got your solvent front here. You measure the distance up to the centre of the substance that you're measuring, and then you divide this distance by the distance that it's moved, the solvent front is, and you end up with an RF value. So the RF value is known as the retardation factor, and your retardation factor will be between 0 and 1. If it doesn't move at all, it'll have a value of 0. If it's up with the solvent front, it'll have a value of 1. But you can't have a value greater than 1 because you can't get the material, you can't get it to move further than the solvent front. So the amount of separation is determined by depends on the length of time the phases are in contact with the solvent. So how long have they been exposed to the solvent and with the ability to dissolve? So if it's too short, then you won't get very much separation between the phases, which makes it very difficult to identify which is which. And if it's too long, then they all end up getting to the top, and you then can't separate them again. So you need reasonably long plates and a reasonable amount of time in order to get the best separation. So sometimes you need to experiment with the solvent that you're using, because some different solvents or different stationary phases will allow better separation of the analyte that you're analysing. So the amount of time that the analyte spends absorbed compared to desorbed is consistent with that particular set of stationary phase and mobile phases. So, for example, if you've got an RF of 0.5, it'll move 2.5 centimetres when the solvent in front has moved 5 centimetres. But if you set up another plate with the same stationary phase and mobile phase, and you leave it for longer, then it might move 5 centimetres, which tells you that the solvent in front will move 10 centimetres, because the RF value will be the same regardless of how long or how far you've left it for. So we're able to confirm the identity of particular substances by running a pure sample of that compound. So it's like a standard. So if we look at this one here, so we can see that we've got 3 different inks, and they're often used if we're dealing with inks. If you've got a sample of handwriting and you want to see what kind of pen it was written with, then you could use chromatography to separate out the colours of the different mixture of the ink, and then you can compare to known inks as well. So in this case, ink 1 has got orange and a blue dot up here, so we can see that this blue dot would be the same substance in ink 1 and ink 2. Ink 2 has got the pink dot down the bottom, they'll be the same, and then the orange one between ink 1 and ink 3 will be the same, and ink 2 is unique in that it's got the yellow dot as well. We can calculate the RF values again by measuring the distance up to how far the solvent has moved, and divide that into the distance that the sample has moved. So we can calculate that the RF value, for example, for this dot here is 0.48, so it'll be the same for that one there. So we can certainly see how it separates out all of the different components of a mixture, and we know that the blue ones, are they attracted more to the stationary phase or the mobile phase? Mobile? Why mobile? Yep, we know that they're attracted to the mobile phase because they've moved the furthest, whereas the pink dots are more attracted to the stationary phase, so because they've moved the least amount of distance. So, a quick summary of thin layer chromatography. So, we've got a solvent that moves over the stationary phase, and it does so by capillary action, and the different components of the mixture that we're trying to separate are either adsorbed or desorbed, depending upon the chemical. That happens at different rates depending upon the chemical or the intermolecular forces between the solvent stationary phase and the analyte, the material that we're trying to analyse. So stationary phase can be fine powder on glass or plastic, so it's something which has got a large surface area, so it comes into, so that the sample comes into a lot of contact with the stationary phase. The mobile phase will need to be polar or non-polar depending upon the material that we're trying to separate or analyse. So if, for example, we are going to do, if we wanted to do the whiteboard markers, if the whiteboard marker ink, for example, has a non-polar ink in it, then would water be useful as a solvent? Why wouldn't water be useful? Yeah, so water is polar, so you would need to use a non-polar solvent if you wanted to deal with a non-polar substance. And then we measure the RF, which is a relative measure of how far the sample has moved, the retardation factor, and it's often used in organic mixtures and plant pigments and drugs. So that's thin layer chromatography. Let's look at gas chromatography. So gas chromatography really works on the same basis. So it's used for small organic molecules that can withstand high temperatures, and they need to be able to withstand high temperatures because we are going to put them through, expose them to heat to vaporize them. Blood alcohol levels are measured using this particular technique. So measuring the amount of alcohol in your blood is done using gas chromatography. So you've got a gas bottle, an oven, a column, which is where the separation occurs, and a detector and a recorder. So unlike the other process where all we're trying to do is to identify different materials, gas chromatography will allow you to identify the amount of a particular material as well, not just its existence. So you inject a sample into an oven and the gas pushes the sample into a long thin column. We'll show you a picture on the next slide. In the column you get smaller particles, and those ones either absorb it onto the stationary phase, and the last ones to leave, the smaller ones are the last ones to leave the column. Say that again. The smaller ones are the first ones to leave the column because they're the ones that get absorbed to the stationary phase the least. Whereas the larger particles take longer to leave the column because they're absorbed by the stationary phase much more easily because they're larger. So in this case, what we're doing is we're measuring the amount of time that it takes to leave the column, and the amount of time that it takes to leave the column is a characteristic for each substance. So we're measuring the RT, which is the retention time, and you can use it to identify the component in the same way that you can identify the RF in thin layer chromatography and paper chromatography. So what you end up with is with a retention time, so over time you end up with a peak height of something coming through to the detector, and then you know the amount of time that it takes for a particular substance to come through the gas column, and you can identify the different peaks, and you can also get an indication as to how much of it is there by the peak height. So this is a setup where we have a gas, which is your carrier gas, which is usually nitrogen, and that's being injected in or being pushed through the coil. We've got a sample injection point, which goes through an oven to heat it up to vaporize it, and we put the material in there so the gas moves through. Some of the gas is more attracted to the substance that makes up the column in the coil, so that material will take longer to move through the coil, and as a result we'll have a longer retention time. So D would be one with a longer retention time, something that's more attracted to the material of the coil, whereas things that are not attracted to the coil will move through very quickly, and as a result of moving through very quickly, they'll come out first, and will be ones with very low retention times or shorter retention times. So the gas chromatography, it's the same kind of idea with our stationary phase. So we've got a special high temperature coating, because remember that this is the coil is in an oven. It's got a fused silica phase or section, and then the stationary phase is bonded onto the fused silica. And these coils can be anywhere from 1.5 to 10 metres long, so they're coiled up so as not to take up much space. So this is kind of a cross-section of the tube that the sample will go through in gas chromatography. So unlike thin layer chromatography, we can actually use gas chromatography to measure the amount of a material. So the way that you do that is by sending through a set of standards. So for example, if we want to test how much alcohol is in a particular wine, we would then send through a sample through standards with different amounts of alcohol. So the amount of time that it takes for the alcohol to go through would be the same. But the actual amount or the peak height would be different. So you'd send a sample through with 5%. You know when the alcohol comes through because it has the same retention time. You measure the peak height, then you do the same thing for 10%, 15%, 20%. They'll have progressively higher peaks. And then you can plot that up on a graph, much like we did with AAS. We had a calibration curve, and then from the calibration curve we can work out the unknown. So this is just an example of a calibration curve. So you've got your concentration and a response ratio. So you measure your response ratio, which is the peak height, and that will give you the concentration of the particular material that you're trying to work out concentration for. So in summary, it's a little bit different to thin layer chromatography. So in this case, the mobile phase is a gas or a carrier gas. You inject your sample into an oven. The gas moves through the vaporized particles, moving through the long, thin column. The smaller solutes will absorb first and elute first, so they'll go out first, the smaller ones. And the R depends on temperature, length of column, and flow rate, as well as the chemical structure. So obviously you're going to need to maintain temperature, length of column, and flow rate if you want to determine different chemical structures. So the stationary phase is a long, thin column. The mobile phase is some kind of inert gas, could be nitrogen, carbon dioxide, or helium. We measure the RT, which is the retention time. It indicates what the sample is. If we look at the area under the peak, then we can work out the amount of substance present. And if we want to know the actual value, then we'll just use a set of standards and create a calibration curve. So it's useful for small heat-stable organic compounds, things like ethanol or for alcohols. It's no good for things that are not heat-stable. So if it's something that if you heat it up, structure breaks down, then it's not going to be very useful for those kinds of things. Last one we're going to talk about very briefly is high-performance liquid chromatography. So this is used for larger organic molecules, and it's used when they're unstable in heat, because there's no oven involved in this style of chromatography. So our stationary phase is much shorter than what it is in gas chromatography, and the mobile phase in this case is a liquid rather than being a gas. And we've got a pump that pumps the solvent, or the mobile phase, pumps it through, and then we inject our sample into the mobile phase, goes through the column and comes out through the detector. And again, we're interested in retention time. We're interested in how long it takes for the compound to come out the other end. And we do the same idea with a calibration curve with a set of standards like we did for gas chromatography. So that's kind of an example of what a curve might look like. So this is retention time down this way in minutes, and we can see that we've got these peaks. So from this graph we can see that the most common substance was this one because it's the tallest peak, and we can see that probably the larger molecules are these ones because they take the longest to move through. And if we've got a calibration curve, we know how tall these peaks are. We can use that to work out how much of that particular substance is in the sample, so we can work out the concentration. And if we're using the same length of the same solvent and the same stationary tube, then the time that the different materials come out will be an indicator as to what that particular substance is. So high performance liquid chromatography is similar in idea to gas chromatography, except for instead of having gas, you've got a liquid phase as the mobile phase. Difference between the gas and the liquid phase is the liquid phase actually does play a role in the adsorption and desorption rates. So you need to have the appropriate solvent depending upon what your sample that you're analysing is. You need a pump to push it through. You have a short column with a particular particle size, and it's extremely small, which allows better separation because you want to be able to really separate out the different substances. You don't want them coming out at similar times. You want clear separation of them. The mobile phase could be water or methanol, or a water methanol mix, or a water acetone or acetone nitrile mixes. So there's a variety of different solvents that can be used there. Again, we end up with a retention time, which indicates what the sample is. The area under the peak indicates how much it is. We can use a standard to get a qualitative idea of what the material is and how much it is. And it's good for larger organic compounds. So that is a pretty quick run through of those different types of chromatography. There's some work there